Hey everyone, my name is Kay Beidel from Rutgers University. I'm one of the co-chairs of the 2018 Ocean Sciences meeting. And on behalf of my other co-chairs, Dick Kraut and Fred Bingham, as well as the program committee, it's my pleasure to welcome you to the Science Plenary Talks. We have a wonderful lineup this morning with Claudi Benitez-Nelson from the University of South Carolina, John Dabiri from Stanford University as our plenary speakers, and we thank them for their time and their contribution to this important aspect of our program. My colleague Dick Kraut will announce John a bit later. Before I introduce Claudia, just a couple of things I wanted to highlight. First, there was an effort this year to make the plenary talks as cohesive and connected around some thematic linkages. And while they have different disciplinary foci, both Claudia and John embrace this idea. And in this spirit, they've been interacting and communicating in the months leading up to the meeting. So keep this in mind as you're looking at their talks, and keep an eye out for those thematic linkages. Secondly, I'm personally very thrilled to have both Claudia and John speak in this setting and with this platform to communicate their ideas. Both of them are inspirational superstars who not only do multifaceted, amazing work, but they have greatly stimulated diversity in our field. Each talk will be followed by a 15-minute question and answer period. So we have a set of microphones in the right, center, and left aisles. So please, if you have a question after each talk, queue up at those. So onward. So Claudia. Well, I'm going to introduce you, Claudia. Hold on. Claudia currently is a College of Arts and Sciences Distinguished Professor in Marine Science at the University of South Carolina. She did her PhD with Ken Bissler, the MIT Woods Hole Joint Program, looking at phosphorus cycling in the Gulf of Maine. After that, Claudia was a postdoctoral fellow and later joined the graduate faculty at the University of Hawaii. And she came to the University of South Carolina as a professor, joined the faculty, first in the Department of Geology, and then later the Department of Earth and Ocean Sciences, where she currently resides. And in that role, she's had many multifaceted roles, but she's served both as the undergraduate director and the director of the Marine Science program at USC. She's been recognized with numerous prestigious awards at her university. University of South Carolina, including the Distinguished Professor of the Year Award. She's had some awards more broadly in the Southeastern Conference and across the professional landscape too, including being named as a National Academy of Science Kavli Fellow, 2012. She received AGU's Saltzman Award in 2014, and she's currently a AAAS Fellow. Her work broadly explores mechanisms that influence particle formation, composition, and flux from the surface ocean to the seafloor, with an emphasis on carbon and phosphorus biogeochemistry and the use of geochemical tracers to interpret that flux. Claudia hails from the Pacific Northwest. She attended the University of Washington, where she majored in both physical and chemical oceanography. Impressive. I was particularly excited in talking to her to find out that she was also a Division I soccer player at the University of Washington and has continued that passion for playing and coaching soccer. As a former Division I soccer and lacrosse player myself and a current coach, that really resonated with me. And I think it serves to further show how dynamic, talented, balanced, and driven Claudia is. So without further ado, Claudia.
Well, thank you so much for such a wonderful introduction. So before I begin, I just really wanted to make a point of acknowledging all of my fabulous collaborators whose work has really expanded my ideas and thoughts and thinking about how the ocean system works. And as Kay mentioned, when thinking about this talk, I was trying to put together this idea of how we move from these oceanic to molecular scales and then back out again. And I think to make sure that I kind of orient us all on the same page, I wanted to show this fabulous figure that Dave Carl put together that really shows these types of scales that we're talking about, both in time and in space. And when thinking about these scales, we can think about them in terms of this kind of biological component, which happens on these shorter time and space scales, moving up into the oceanic conveyor belt, ocean mixing, and then finally thinking about geologic time and the processes of erosion and runoff that then influence all of these scales put together. And for me, one of the grand challenges is how do we link these scales? How do we bring them all together? And so that arrow, of course, is a straight line, but in reality, it's much more complicated. So teasing apart these interactions, that to me is really where it's at. And so I'm a biogeochemist, and as biogeochemists, we tend to talk about the big three, and that's carbon, nitrogen, and phosphorus. And to be even more forthright, we tend to have our favorite elements. We tend to be carbon people, or nitrogen people, and a few phosphorus people. Thank you. Right? And we tend to maintain our own little spheres. And that's actually understandable. Because if you think about these cycles, and so here I have a nitrogen cycle and a phosphorus cycle. I didn't even bother with the carbon. Okay? You'll notice that these cycles are incredibly complicated. These elements have different speciation states. They go under reduction oxidation reactions. They change their phase from solid to liquid to gas. And even though we've been studying these particular elements for decades, there are still aspects of them, the anammox cycle, this new phosphorus reduction cycle, that we're only now beginning to discover and fully understand. So the question then becomes, when you have these very complicated cycles, is how do you put them all together? How do you distill this down in such a way that you can begin to make progress on a bigger scale? And so for that, I really want to talk about Redfield. Now, Redfield was this amazing individual. And what I really like about Redfield was that it was his attempt to really think about how to put these very broad, diverse scales together and link them. So for those of you who don't know about Redfield, I know most of you do, but for those of you who don't, what Redfield really posited, or what he really discovered, is that when you looked at these larger organisms that lived in the ocean, and you measured their carbon to nitrogen to phosphorus ratio, it was amazingly distinct in that it was 106 to 16 to 1. This was the average ratio that you measure across the oceans. And that when he looked at nutrients in the water column, this ratio of nitrate to phosphate, that was also 16 to 1. And so what Redfield posited and put forth is that the environment fundamentally influences the organisms that grow, and the organisms that grow in the environment fundamentally influence the environment itself, right? And that this dynamic interplay was what really kept this elemental ratio or stoichiometry at this 106 to 16 to 1. And this idea, this thought has really permeated our thought processes in biogeochemistry, to the point that when we measure carbon or nitrogen, we'll use the Redfield ratio to get to the phosphorus. If we see a ratio where the nitrogen to phosphorus is 16 to 1 and say we have a little bit more nitrogen, right, so that ratio is higher, we'll say, it must be phosphorus limited. Or if the ratio is lower, it must be nitrogen limited. And we'll even use this ratio when we talk about large-scale biogeochemical cycling and these nice interactive biophysical models where we'll just kind of transform, one element into other using Redfield. And so this idea has become so pervasive that even the trace metal geochemists are getting into the action, right? So this is a plot of mean dissolved seawater ratio of trace metals to carbon. And then at the cellular ratio, there's the trace metal to carbon ratio, and you can see that nice plot, again with this idea that trace metals that are in the dissolved phase versus what you see in the particulate phase are interrelated. So I want to take a step back now and really think about Redfield ratio and how this really works in kind of this large-scale global ocean idea. So this is a fabulous figure from Gruber and Deutsch, and what they've done is they've plotted nitrate versus phosphate. And those little circles that I don't know that you can see very well, that's Redfield's original data. And then all those little points, those are data from the World Ocean Circulation Experiment. And it's from the Pacific Ocean, the Atlantic Ocean, and the Indian Ocean. And these are all these waters that are sampled below that upper few hundred meters. And check out that linear relationship, right? For you graduate students in the audience, it doesn't get any better than this, right? It's 16-to-1, right? It's awesome. And what Gruber and Deutsch put out when they were having the discussion in this paper is this idea that when you get too much nitrate, the oceans will change their redox chemistry, right? And they'll cause denitrification, and that's a way to get nitrate out of the water, convert it to N2, and to bring that ratio back to our wonderful Redfield relationship. At the same time, when you have too little nitrogen in the water, you have these amazing organisms, cyanobacteria, that'll fix nitrogen from the water column, convert it to their nitrogen needs, and again, move the nitrogen system back to that Redfield ratio, right? Okay, that's a little nitrogen-centric. Let me just put on the phosphorus point of view here and say that those same processes influence phosphorus as well. So when you get into these anoxic, no oxygen waters, you can reduce iron oxides, you let phosphorus back into the system. Okay? At the same time, those cyanobacteria that can fix nitrogen, they'll take up excess phosphorus, right? So again, these processes moving the system back into this Redfield kind of 16-to-1. So that's dissolved nutrient stoichiometry. Things get a little bit more interesting when you start moving to particles, right? When you start thinking about how these cells and how these organisms exist in the water. So this next figure is kind of a global view of suspended particles. And I apologize for all the biologists in the room that I call their phytoplankton and bacteria in the surface ocean suspended particles, but so be it. Right? And what I want you to notice, and I know it's a little complicated, and I'm going to try and explain it. So on this bottom axis is this N to P ratio. And this orange line now, that's that Redfield 16-to-1. And if you look at this bottom figure, you'll see there's nitrate concentrations. And when you have high nitrate or high nutrients, really, your N to P ratio is less than 16. It's about 5-to-10. When you have low nutrients, right, your N to P ratio shifts. And now it's significantly higher than 16-to-1. It could be as high as 30. And so if you think about this as the oceans as a whole, what you realize, or what Martini et al. Showed, is that you tend to have high N to P ratios in these oligotrophic waters, and you have lower N to P ratios when you have waters that are nutrient-rich, so at the poles, versus what you see perhaps at the equator. And what they attributed this to was these different communities of organisms that are living in these waters. So that when you have high N to P ratios, you guessed it, you're dominated by these organisms, these cyanobacteria that can fix that nitrogen from the atmosphere, use it for biomass. They tend to occur in these really stratified, warmer systems, not all, but that's when they tend to occur. And then when you have low N to P ratios, you have these diatoms, right? They have these nice, they're big, they're juicy, and that's where you have the high nutrients. So those are suspended particles. What happens when you take this material and you make it bigger and you now allow these particles to sink down into the deep ocean, right? Because it's these particles that are sinking into the deep ocean that are fundamentally going to influence that dissolved nutrient phase that I was talking about earlier. So here's the sinking particle stoichiometry. And again, there's nitrogen and there's phosphorus. And there's that beautiful Redfield ratio of the 16:1. Now, this is data taken from a variety of regions, the Bermuda Atlantic time series, right, in the Sargasso Sea, Hawaiian Ocean time series in the Pacific, the Carioca Basin, the Gulf of California, and the Santa Barbara Basin. And what I'd like you to notice is that you tend to have these higher N to P ratios in these oligotrophic waters, right, of Hawaii and Bermuda, and you have these low N to P ratios when you're kind of in these more nutrient-rich ecosystems, which of course, you guessed it, right, are dominated by these kinds of two end members of communities. The cyanobacteria on the one hand, we knew Bermuda has a lot of cyanobacteria, the Hawaiian Ocean time series has a little bit less so, and then at the other extreme, the Santa Barbara Basin, which we know is very diatom dominated, and then kind of these two other time series that are kind of in between. So now, Redfield, on average, 16:1, but we clearly see these large-scale regional differences in terms of the stoichiometry of both the suspended and then how this material sinks out of that upper 200 meters. Now, just for kicks, I want to take it even a little deeper than that, okay? So one of the grand challenges, I think, in terms of trying to understand this information is that we are so data limited, okay? We just don't have a lot of sites that are looking at suspended and sinking particles over long time scales. But one of the sites that we do have is a place called the Carioca Basin, and I love it because it's been out. These are samples that were collected for the past 25 years. And what I really like about this ecosystem is that the data shows there's five different sediment traps that are placed throughout the water column, right? So you have these 5 different garbage pails that are essentially right up in that surface ocean, and then you can actually see how material is making its way right through that upper 1,000 meters where all the action is. And in many other places, what happens is you kind of have that 150-meter garbage can, and then you got one at 2,000. And you kind of hand wave about everything that happens in between, right? So here it's pretty cool, right? You kind of get the whole water column. And because it's got 25 years of data, even though it's really variable, you can start to kind of tease out some patterns. So again, here's depth. Here's that N to P ratio. There's the orange, N to P for Redfield, 16:1. And what I've plotted here is blue is kind of this upwelling diatom-dominated system. And then in the red is a non-diatom-dominated system. And you'll see I've kind of got some cyanobacteria there, but not really. And that's kind of because we haven't really noticed a lot of cyanobacteria until relatively recently in the basin, right? So we think they're kind of new and moving in. So there's a few things I really want you to notice about this one, okay? And that is, if you look at that kind of upper 150 to 275 meters, right, you'll see there's a really large-scale change in the N to P ratio of the particles that are sinking out. That N to P ratio really increases. You're getting preferential remineralization of these kind of phosphorus-containing compounds. But perhaps what's really surprising about this is that this is happening with these diatoms, right? That we think of these fresh, juicy kinds of organisms, right? But we kind of think of them as sinking pretty quickly, right? So they're kind of like these little bombs that are making their way out. So we're seeing really rapid remineralization. And then during the rest of the time in that upper kind of 300 meters, we're not seeing a whole lot of action. I mean, it definitely increases, but not nearly to the same extent. And then when you get into kind of that 400 meters and below, these are these anoxic waters. Carioca Basin is anoxic. You don't really see any change at all, right? So it kind of turns, and then it comes straight down. And I'm going to come back to these anoxic waters in a minute, or maybe a few minutes. But I just want you to pay attention to that. Because at the end of the day, this difference now and how these N to P ratios change as these particles are moving down through the water column is fascinating. Because it means that not only are we seeing very different material at the surface, be it these diatoms or the cyanobacteria, but that this material is fundamentally different as it makes its way down, right? And so for the diatoms, I love M&Ms. And I hear that Colin Rensler loves peanut-covered or chocolate-covered M&Ms, chocolate-covered peanut M&Ms, sorry. Right? So it's really good, yummy stuff. And now we have this set of organic matter that as it's making its way down to the water column, perhaps for whatever reason, is not particularly juicy, not particularly able to be degraded. And in my mind, those are Brussels sprouts, because I just hate Brussels sprouts. I know some of you like them, but there's only a few of you. Okay? So now we're talking about stoichiometry and the composition of these particles, and we're talking about them regionally. We're talking about as this material now sinks through the water column, right, the composition. And the reason why I think about this is because ultimately the material, again, that makes its way down into the deep water influences the nutrients that are there that could participate in this kind of long-term mixing of the oceans. So this is a figure, this is a longer kind of temporal view of nitrogen to phosphorus stoichiometry by Planowski, and it goes back 4 billion years. There's some hand waving there, I get it. But it's cool. And it's cool because even though we know the Earth was a very different place 4 billion years ago, you can see that this idea is that we have these ecosystems, these oceans that are dominated, again, by those high N to P organisms, cyanobacteria. And about 700 million years ago, we see this transition. We see these N to P ratios going down. We're seeing the rise of these eukaryotic organisms, these diatoms, that have this ratio of 106 to 16 to 1. And I put this up here because I think it's really fascinating to think about how these ratios have now changed over these longer time scales, because we can start to really see these changes now on kind of the decadal time scales, particularly at the time series station. So here's the Hawaiian Ocean time series station. Those are those same sinking particle fluxes that I showed a few slides earlier at 150 meters. And there's the N to P ratio of the sinking particles. And you can see there's my beautiful Redfield orange line there, about 16. And so, of course, yes, all those sinking particles have a higher N to P ratio, right? But look how it's changed over time, particularly over the last 17, 20 years. That N to P ratio is slowly but surely increasing. And at the same time, this is a system that we think is increasingly dominated by these cyanobacteria, right? These nitrogen-fixing organisms. And so this is really thought-provoking as we think about then how we're going to move into the future, right? This idea that we're going to have more stratification, more warming as temperatures rise, right? Expansion of these oxygen minimum zones. And so I put this out here because in some levels we think, oh, it'll keep going, and then eventually we'll run out of phosphorus, and then we'll flip right back, and we'll have this kind of amazing biogeochemical feedback loop that's going to keep everything in line. Well, I'm not so sure that's true. And so one of the questions that I've been really thinking about then is how high can those N to P ratios go? How high can they go? What's the composition of this material? What's fundamentally changing? Can we model it? Can we predict it? And so to do that, that requires that we fundamentally go beyond kind of these bulk measurements of the elements. And this is me saying this when we really don't have a lot of bulk measurements to begin with. We really don't, right? So we don't have a lot of bulk sinking particle fluxes of nitrogen and phosphorus and carbon. And now I'm going to tell you we got to go to the molecular scale. And so when we think about then the molecular scale, I'm going to tell you right now, I'm really going to focus on the N to P ratio and those compounds that contain nitrogen and phosphorus, just because the variability in that ratio is so much greater than the ratio that we observe for carbon and nitrogen. So let's get molecular. Let's go deep. And for all you biologists, I'm really talking about chemical compounds now. Sorry. Right? Don't freak out. I know, you guys all just saw me put up these chemistry compounds. You're like, oh my God. No, it's okay. It's going to be okay, I promise. I tell my students that. They don't believe it either. So let's talk about the biological diversity of nitrogen and phosphorus that occurs in the cell, right? We kind of have these classic compounds of ATP, DNA, RNA. They're pretty varied. They have all sorts of bonds associated with them. But I'm really going to focus on two classes of compounds. And the reason why I'm going to focus on them is, frankly, because they're great compounds. If we're going to budge that N to P ratio, then we should really focus on compounds like polyphosphate, which has a lot of phosphorus and no nitrogen, right? That's a great way to really change that N to P ratio. The second class of compounds I'm going to talk about are phosphonates. They have this strong carbon to phosphorus bond. And again, they seem to have fairly little nitrogen. The third class of compounds, which are awesome, the phospholipids, I'm just not going to have time to talk about. Right? So I'm just going to really focus then on the polyphosphates and the phosphonates. So one of the big issues, though, with looking at things at this kind of chemical perspective and getting down to specific compounds is that it's a tough nut to crack, particularly when there's nitrogen and phosphorus involved. The whole idea of looking at a compound, extracting it out, you've changed the compound, right? And so how do we do this, right? And so that's really been a big area right now, so is how do we look at the molecular scale of all these different compounds without fundamentally changing the material that we're interested in looking at to begin with, right? All right. So I'm going to start with polyphosphates, all right? And I'm going to focus on one of our N members, diatoms, right? So diatoms will be one of our N members, and we'll do cyanobacteria as the other N member. So this is a diatom. And what this technique is a simple Dabiri stain where you've just stained it, and you can see this nice fluorescence here from the polyphosphate inclusions. It's those little, see those little green circles there? And in fact, if you look up on the side there, you can see the little greens, the work that we're just doing with Julia. Now remember, polyphosphates, these are just phosphate, 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 phosphate. There's no nitrogen, right? And if we do some significant chemical extractions and we really pull it out, we can see that if we plot opal versus polyphosphate, we kind of get a straight line, right? So we can see that polyphosphate is a component of these diatoms, and it's about 10%, right? So the Dabiri stains are great because it shows that there's polyphosphate in there, but it doesn't really tell us the actual amount of polyphosphate that's in there. We can do these really hardcore chemical extractions, and we can get to the amount of polyphosphate in there, but it's tough, okay? So we have polyphosphate in diatoms. It's about 10%, right? That N to P ratio is low. Hey, at least some of that's got to be due to this polyphosphate, right? There's no nitrogen in it. Okay, that makes some sense, right? That's good. We got it. One of the interesting things then is what happens then to this material as it sinks. And I'll just say to you, one of the fascinating results of this work was that whenever we looked at the polyphosphate and as it made its way through the water column, even though phosphorus in general is remineralized, the percentage of polyphosphate is always the same. The percentage of polyphosphate to the phosphorus pool is always the same. And that was really surprising because polyphosphate, let's be honest, that's pretty yummy stuff, right? It's good stuff. I mean, if I were an organism and I was going to go after a phosphorus compound, I'd be going after the polyphosphate. And what we found after a little bit further investigation, now this is now we're on the third technique for measuring poly-P, right, is that when we took a really closer look at this material as it's sinking from the surface ocean down into the deep ocean, and you can see there's these little inclusions and those little red squares, right? You can really blow up and look using Zanes to kind of get an idea of the spectrum and the mineralogy of that material, is we found that this polyphosphate was converting into these apatite forms as it sank through the water column. Okay, right? So now we're like, okay, we know that polyphosphate is a significant component of these diatoms as they sink through the water column. We're not seeing preferential removal of the polyphosphate, right? So that means that when we see these large-term changes in the N to P pool of the sinking organic matter, it's really everything that's being remineralized, right? We're having equally amounts of this nitrogen and phosphorus. The polyphosphate seems to be okay. And then as it makes it down, it kind of stays there. So let's go to our other end member, Cyanobacteria. Let's look for polyphosphate there. And I'm just going to tell you right now that we found polyphosphate in Cyanobacteria. This is, I think, the fourth way that we've now measured polyphosphate. This is using NMR, and that little peak there, that edge, that's a polyphosphate peak. So this is some work we're doing with Sonia Dyerman and the Sargasso Sea. And kind of the end result of this is that cyanobacteria can have as much as 25% of their phosphorus be in this polyphosphate form. Let me say that again, 25%. I have just told you that cyanobacteria have these really wicked high, can I say, yeah, wicked, okay. These really high N to P ratios, right? 25, 30, and then that phosphorus is composed of 25% polyphosphate that has no nitrogen in it. So that means the remaining component of these cyanobacteria must have even higher N to P. Surprising, right? Shocking in many ways. All right, so what happens to the poly-P as this material sinks through the water column, right? So this is some great work by Martini et al. They published in 2014. And this is really two systems here. So we have this temperate western North Atlantic, it's higher nutrients. And then we have my favorite place, the Sargasso Sea, right, which is low nutrients and dominated by these cyanobacteria. And what I really want you to look at is that bottom set of figures, okay? And what you'll see on both sides is that this is the poly-P to total phosphorus ratio of material as it sinks from the surface ocean through that 150 meters, okay, that upper water column. And what you'll notice is that the poly-P is about 10% of the total phosphorus pool. I know that those axes aren't labeled, but it's about 10%. And that you don't see, even though phosphorus declines in general, hey, that poly-P to total phosphorus ratio stays the same. It's exactly similar to the data that we showed previously, right? It's always good to get back up. But now let's look at the Sargasso Sea with these sinking cyanobacteria. And number one, there's that poly P to phosphorus ratio. It's about 25%. But now let's look at what happens to that poly P relative to phosphorus as it sinks out of that upper 150 meters. That poly P is gone. It remineralizes so quickly. So by the time you get down to 150 meters, there's hardly any of it there, right? So now, this is yet another mechanism of how you increase this nitrogen to phosphorus ratio of these very specific group of organisms as it makes its way down from the surface to depth. Okay. There's your stoichiometry. All right. Now, I want to continue, and I want to really think about then kind of this longer term. Here's the sinking flux at the Bermuda Atlantic Time Series. I just downloaded it from their data repository. It's awesome that they put their data online so all of us can access it. Hint, hint. This is an N to P molar ratio. And here's time from 2005 to 2017. This is all of their N to P data. There's my favorite orange N to P line of 16 to 1. Again, notice, right? The ratio is always above that Redfield. But check out what's been going on in the last 5-10 years. Those N to P, that ratio has really just taken off. And I think that's real data. Boom, once again, these increasing cyanobacteria. So this is fascinating to me because I really want you to pay attention to that scale, that molar N to P ratio, right? We're talking about 25, 30. We are now up at about 300. How high can those ratios go? How high can they go? I don't know. Right now, I'm thinking they can go pretty high, right? And again, remember, this is the material that's feeding that deep ocean, right? All right, we'll talk about one more compound. I want to talk about phosphonates. So phosphonates are these compounds that have really strong phosphorus to carbon bonds. All right, now this is NMR. And you can see there's that phosphonate hump. And what's really interesting about phosphonates is that we've known since that real seminal work by Lauren Clark-Kolowith and Ron Benner and Ellery Engel, that they are a significant component of the dissolved organic phosphorus pool. They're about 6 to 10%. Okay. But what's been fascinating about this is that only until recently could we figure out where they were coming from. So we have these phosphonates, and for years we just were like, I don't know where they're from. We don't know how they got there, right? We don't know what they were doing. Subsequently, Sonia Dyerman and her colleagues in Metcalf realized that, you guessed it, cyanobacteria, right, had these ability to break down these phosphonates and use them for their phosphorus needs. But again, where are they coming from? So this is work we were doing with Sonia Dyerman. We were looking at the phosphorus composition of a variety of cyanobacteria. And boom, there they are, right? We found them. This is in Trichodesmium erythraeum. This was a culture study. We found that they were about 10% of this phosphonate pool. Now, for us, we were like, wow, this is great, right? We finally found where these phosphonates might be coming from. But then we have the sickening realization that when we looked in the other cultures, and we looked at these organisms as they were in natural populations, we really couldn't find them, right? So we saw them, we can see them in this one culture, but nowhere else. And since that time, there's been work that has shown then that there are many organisms that have the ability and the genetic code to synthesize these compounds. And yet, in my mind, I was just, gosh, it was this really puzzling question, and it just bugged me, right? Here we have these compounds, they're in the dissolve phase. We see them in some of these cyanobacteria, but why don't we ever see them? And then Van Mooy came along, right? And what Ben Van Mooy showed in 2015 was actually a number of really cool things. But one of the things that he was able to demonstrate using yet another technique, right, is that there are phosphonates being produced by cyanobacteria. This work is in the Sargasso Sea. And not only are phosphonates being produced by these organisms, but so are these other kind of funky reduced compounds, right? Phosphites. Reduced phosphorus, really? But they were. But here's the take home. These compounds are not only being produced, but they are turning over at a rate of about 1 times 10 to the 11th moles of phosphorus per year. That is the same order of magnitude of the phosphorus that enters the ocean on that time scale. That's right. They are producing these phosphonates and using them as quickly as they get them. Boom. Makes me happy because now I finally understand why I can't find them, right? Because remember, when you're looking at organic matter and you're only looking at what's there, right? You're not looking at what's been used. But fundamentally then, what does this mean? It means that here are these organisms that, again, the phosphorus is turning over so rapidly that allows that N to P ratio to get pretty high, right? Because phosphorus turning over could be really low, but it's turning over really fast. So again, how high can that N to P ratio go? And I have some time, so just to throw all you carbon people a bone, okay? Phosphonates are really interesting because, oh yeah, they impact the carbon cycle too, okay? And they do so very directly. So some initial work by Angel White and then Dave Carl and now being really some beautiful work by Dan Rapetta is really showing that there are these organisms out there, the cyanobacteria, that in the process of producing phosphonates and breaking them down and producing phosphonates and breaking them down and breaking these incredibly strong phosphorus to carbon bonds, they produce methane. And some of that methane, yes, can escape into the atmosphere where it's a greenhouse gas and 20 to 25 times more effective than carbon dioxide, right? So what does this mean? What does this mean? So this is a global biogeochemical model that was put forth by Tang et al. In 2016. He didn't have a lot of data, but he really did the best that he could. It's a great model, focusing on carbon to phosphorus, of course. And so I've converted it to nitrogen to phosphorus using, guess what? Redfield. Okay, and what I want to show you are these dark green, those are those cyanobacteria, high nitrogen to phosphorus dominated ecosystems. And then there's the light green, those are, the diatoms, right? And as we think about this, and as you look at this figure, what I want you to think about is how expansive can those dark green areas grow? Right? How can these biomes shift? Are they going to expand? How high can those N to P ratios fundamentally go? And what does that mean then as we think about how our ecosystems and the biogeochemistry is fundamentally changing in our oceans as a whole? And are we moving into a new kind of steady state? Are we slowly making our way into hitting a tipping point? I'm not so sure I know. What I do know is those cyanobacteria, those N to P ratios, they can get high. And from looking at the molecular scale, they can go even higher. So I want to end with this idea that, Redfield had it right. By looking at these elemental cycles and how they interact with one another, really can begin to tell us something about ecosystems and how to view things, I think, in a little bit different way. And it's an exciting time to be an oceanographer, to be a chemical oceanographer, a biogeochemist, because we're fundamentally developing these new tools that we can really dive down now into the molecular level. What's fundamentally going on in these systems and in these smaller scale that then influences the larger scale. But of course, it means that we have to measure these elements at the same time. And not only do we have to measure these elemental pools at the same time, but we have to do them in a variety of environments. Not just the Sargasso Sea, right? Not just Carioca, but region, depth, oxygen level. I think it's critical. And because we're at the Ocean Sciences meeting, we had about 1,600, I think, abstracts submitted to the meeting. How many? 1,600? Okay. 4,000. Oh, gosh, excuse me, 4,000. Should have looked before I said anything. Okay, how many of those abstracts do you think mention the words carbon in them? Oh, come on, it's like my freshman class. Come on, people. 1,000. It's because not bad, Dr. Bronk. 830. How many of these abstracts mention the word nitrogen in them? Someone said 16, sorry. 240, right? That's our C to N ratio right there of about 3. How many of these abstracts mention the words phosphorus in them? Oh. Well, yeah, take that, people. We're coming on strong, right? That N to P ratio, that's about 67. How many abstracts do you think mention the words like carbon to phosphorus, nitrogen to phosphorus, ratio, Redfield stoichiometry? I didn't put C to N there because you get N as a byproduct of carbon. That's cheating, right? So I don't see it in. How many? $20. Okay, 20. That's what I said. Wow. Right? Sounds really low. But I put this up here because really, I think we got to get out of our boxes, okay? And really think about these elemental cycles a little bit more, even though I know it's so difficult because our own individual cycles are so complicated. So with that, I'll end. Thank you very much. I'll end with my favorite phosphorus nerd joke. All the phosphorus people out there will get it. Thank you very much. Remember not to take that from you. Questions? Oh, sorry. Yes, to my left.
If you give 3-hour lectures like that a week for a semester, you probably explode a lot of skulls. And that's really nice.
I have some former students in the audience. They can tell you how they, yeah. Oh, he raised his hand. Look, he's like, yay. He's the guy with the big mohawk. You can't miss him.
I'm sure they can. I'd like to take the geo out of biogeochemistry for a moment.
Oh.
And think about biochemistry, because
Yes.
that's what the organisms that you're talking about do for a living. And phosphorus and polyphosphate are very interesting because polyphosphate has a function, and that is to provide phosphorus for RNA and DNA and other biological molecules. And in your Sargasso example, your sediment trap is in the photic zone. And I'll bet that your trichodesmium was collected near the surface. And there's a possibility however distant, that phosphorus is being accumulated from, for example, Saharan dust. And that as the trichodesmium becomes happy with their phosphorus quota, that they start to sink down, which looks like sedimentation, but actually, which for them is just an opportunity to live somewhere where there's enough light and there's other stuff going on. So each time those cells divide, they get half as much, but they use the polyphosphate preferentially to make the next round of RNA. So I'd just like to ask how much you have considered that and who you're working with in physiology to take those things into account.
So the short answer is absolutely. I've been working very closely with Sonia Dyerman. These are questions that we think about quite a bit, right? What are the genes that are being upregulated and downregulated in response to the environment that they're in? As these organisms are moving up and down in the water column and changing their light regime and dividing, how are that material being subdivided? And I think, in my mind, to some extent, there's a couple of things, and that is, polyphosphate is a storage molecule. It is meant to be used, right? So it makes sense to me that you get these, that you get trichodesmium that might have polyphosphate. I'm still a little shocked about the amount of it, though. Come on, 25%, that's a lot, okay? And that, yes, it's getting broken down and it's being utilized as these, as they're growing and as they're, participating in their life cycles. For me, in some ways, though, it's this interesting contrast that here you have these cyanobacteria, really high N/P ratios, and then they have this component of phosphorus that doesn't have any nitrogen in it, right? So that means that the rest of the cell has to have really high N/P ratios, right? So I guess that's kind of the first point. And then the second point is now you have these diatoms that also have polyphosphate and are using them for exactly that, energy storage, and yet we don't see that same kind of remineralization, possibly for other geochemical reasons, right, versus biological. And so it's that dichotomy and that contrast that is the most fascinating to me when we're looking at these end members and how these various elements are being cycled. But you're right. In the center, please. And I'm sorry, I can't see you because of the light.
Oh, very nice talk. Thank you so much. This question is motivated by your dark green, light green map. You don't have to bring it back up, okay. But and also in several of your previous slides, you connected some structures with the physical environment, say upwelling, favorable regimes, et cetera. With some of these dramatic changes that you have seen in places, are you thinking you might be seeing initial indications of changes in the physical environment which might otherwise be unobservable if you were to just go out and make measurements of the physical environment?
Absolutely. So the question is, I guess you've all heard it, but what's the change in the physical regimes that are happening, particularly at these time series sites, right, which are so valuable because when you go out, and you go out every, in this case, every month for years, right, you get to have this idea of, whether what you're seeing is real or, or just an event. And so I will say yes, right? Hawaiian Ocean time series, Bermuda Atlantic time series are seeing increasing stratification, reduced mixing, warming temperatures, changes in community structure as a result of that I think is fundamentally influencing those ratios. I will say that we're seeing these same kinds of changes in the coastal ecosystems, right? I didn't really spend a lot of time, but the Carioca Basin, right? We've noticed it used to be this really diatom-dominated, community, and now we're starting to see more cyanobacteria, nitrogen fixing coming in. The Santa Barbara Basin is increasingly diatom-dominated, huge diatoms, and toxic ones, by the way, they're now pervasive in that basin. So there's certainly changes in how nutrients are being transported into these areas and how they're making their way into the surface ocean or not. that's really influencing these communities. And and that's kind of the big key, right? That's what really links kind of the large scale to the small scale, is these physical changes and mixing that kind of puts them all together. So you're absolutely right. Physics is important. Physics is important. Yay, physical oceanographers rock. All right. In the center.
Great talk. If I remember it correctly, the original Redfield data was based on net plankton.
Yep.
So it was missing all your, at least unicellular cyanobacteria, and also the heterotrophic bacteria in the ocean. And I was wondering if, taking into account those organisms, given that they have a little bit of different Redfield ratios, or not the Redfield ratios, they deviate from Redfield basically, and there are huge potential for impacting biogeochemical cycles. If the pictures change, or what are the implications there?
Right. So excellent point, and I really did simplify things quite a bit. So yes, Redfield's original data was looking at net tows, right? So 64 micron, like big stuff. And in my mind, as I think about kind of these larger scale communities, is what are they eating, right? What is the food source that they're getting? And as this material makes its way from the surface and now goes into the mesopelagic, supporting those, the benthic food chain, are we now going to start seeing influences on these larger organisms as we change both the magnitude of the particles that are sinking out and the nutritional quality, right? And that's why I was talking about kind of my chocolate-covered peanuts analogy, and the Brussels sprouts, is that I really do think that that's right. There's going to be fundamental changes in the food that's now going up the food chain, and is that going to now influence the varieties of organisms that live there and how they forage, right? Now, not everyone likes chocolate-covered M&Ms. I know there's a few of you out there who don't like that. In fact, there's probably some of you out there who prefer Brussels sprouts, right? So my end to you is that, the Brussels sprout eaters, they might do better, And I don't know that we have seen kind of large-scale community changes in the Carioca Basin. We're starting to see large-scale community changes. Jeff Drazen, Debbie Steinberg are starting to kind of see these in the Time series, right? And so I'm just rambling right now, but yes, I think they're related, right? And how does that food make its way? It's a really interesting question, and biology is complicated. That's why I'm not a biologist. I might go back to school. I would have studied cyanobacteria. No, never mind. I take it back. But I love those organisms. They're really interesting. Okay. Yes, in the center. Sorry.
Okay. I want to say I loved your talk, and it's talks like that we all need in this era of Trump. This is the way we need to present science to our colleagues and to students and to our brothers and sisters and mothers and fathers. This is the way science needs to be presented. Anyway, thank you for
Thanks.
that.
I'm great, good. No, you're going to ask me a question? Oh, we could just end that way.
Okay,
we're good.
But now I got a science. Now I got a science question. Okay.
Sorry.
How important would it be if you could measure the loss efficiency of the nutrients from the euphotic zone, for example, or just below the euphotic zone, or the retention efficiency of nitrate and phosphate that you've mentioned so much? Is this of interest in your particular work?
So the short answer is absolutely. And okay. I'm really excited by the autonomous measurements that are now coming out where we can get really fine scale examples of kind of the nitrate profiles that we're seeing in the water column and the oxygen. Not really there for phosphorus yet. Come on, people.
Okay.
But we're getting close, right? And I like this because if we can do enough of these temporal profiles, yes, right? As a geochemist, we can start to look at the nitrate, phosphate, how these nutrient concentrations have changed over time, their relative ratios. We see that in kind of enclosed basins. Again, I'll just use the Carioca Basin because we've been there so long. But we have measurements of those nutrient concentrations going back 60 years. And so you can really see how it's changed with time. As you get to the open ocean, of course, it's harder. It's a big place. You have inputs. But if we can do these kinds of fine scale measurements over and over and over again, absolutely, right? Now, does that mean, do I think we still have a limit on those cyanobacteria, that NDP? I mean, go ahead. Let's change those nutrients, because the Sargasso Sea, we have phosphorus concentrations down on a nanomolar. Hawaiian Ocean Time Series, it's 200, nanomolar if you look at phosphorus. So, and yet, right? We have these same kinds of organisms that are existing in both areas.
And yet, you're still working with the statics. What I'm talking about is the dynamics. Basically, the changes in which the nutrients can be retained in the euphotic zone or just below.
Right, and so that's what I mean, is if we can measure them over and over and over and over again, you can start to look at trends.
Yeah, but you don't have to measure it over and over again. All you have to do is measure the enzymes that are doing, talking about going to biochemistry, and that, no, really.
Yeah.
The enzymes control those rates, and yet you can make them at a static, in a way you measure a static property. So if you measure the respiration in the water column, divide it by the carbon flux, you've got, and convert it to Redfield, you've got nutrient retention efficiency. It's just a little, this is a little thing outside the USA. This is from the Canary Islands.
No. I agree with you. The enzymatics are really fascinating. But I will say that, again, as you've mentioned, biology is complex. And the varieties of enzymes that are out there and how they break down and how they form organic matter is so diverse and so responsive. Right? We know that we can see evolution. I think it's a great idea, and I really need to think about it more. But my concern then is the plasticity and how these varieties of organisms express those enzymatic reactions across kind of the diversity of environments that they're in.
That diversity is there, but there's also a uniformity. Anyway, we can talk about
Okay,
this.
yeah, we'll talk.
It turns out you and I have the same background, but in reverse. UW, MIT, it's interesting.
Go Dogs!
Let's give a warm thank you for Claudia. Very nice talk.
That was fantastic, and I'm sure that our next speaker will be just as good. My name is Dick Kraut, and it's an honor for me to introduce Professor John Dabiri as our second speaker in this science plenary. John was raised in a small town in Ohio and went to Princeton University, where he wanted to study helicopters and rockets. There he earned a degree in mechanical and aerospace engineering. Then he completed a master's in aeronautics and a PhD in bioengineering and aeronautics at the California Institute of Technology. He began his professional career as an associate at Caltech and became a professor at 24 years old. By the time he was 28, he was tenured. He was the director of the Center for Bio-Inspired Wind Energy, and during his final year there, he was the dean of undergraduate students. He was also a senior fellow in the Center for Turbulence Research at Stanford University before joining Stanford as a full professor. And there he is currently a professor of civil and environmental engineering and mechanical engineering. In addition to numerous research grants from the National Science Foundation and the Office of Naval Research, he's also received grants from the Gordon and Betty Moore Foundation, the Tomcat Center for Sustainable Energy, and the Woods Institute for the Environment. After he delivers our plenary, he'll be providing the J.M. Burgers Lecture in the Netherlands, an invited lecture at the World Congress of Biomechanics, and a Sears Lecture at Woods Hole. Although trained as a mechanical engineer, he began his career investigating the fluid dynamics of jellyfish by injecting dyes into the water around a swimming moon jellyfish and watching how the vortices of color were shaped by the jellyfish's pulsating bell. These vortex rings travel downward towards the tentacles, adding to the forces produced by the jellyfish's pulsating bell, providing more thrust at no extra energy cost. He then developed better ways of studying these dynamics. Instead of colored dyes, he seeded aquarium tanks with tiny glass beads and then illuminated these with green lasers. The lasers tracked the movements of the beads as they flowed around the swimming animal, and then he developed algorithms using these patterns to deduce how the creature was changing the water pressure around it. And there are substantial ocean mixing implications from this type of research. In a jellyfish, the shape, size, and speed of these rings inform you about how efficiently they travel. The principles of jellyfish propulsion have been extended to determine how efficiently the chambers of the heart pump and to develop more efficient underwater vehicles and wind farms. On a lighter note, John related to me that during his 10 years working with jellyfish, that he avoided being stung by them. But during a photo shoot for Popular Science magazine, he was asked to hold a jellyfish. Being a beach photo shoot, John was wearing shorts, and although he had gloves to hold the jellyfish, the tentacles were long enough, they extended down to his legs, and from all accounts, very painful experience for him. John's research focuses on science and technology at the intersection of fluid mechanics, energy and the environment, and biology. The title of his presentation today is Biological Propulsion in and of the Ocean. Please welcome Professor John Dabiri.
Thank you. Thanks for the introduction. Thank you very much. It's a great pleasure to be here and to share with you a line of research that we've been pursuing for the past dozen or so years. This work was really inspired by one of my heroes in science, Walter Munk, who of course is well known to this group. Around 2005 or 2006, I was a new assistant professor down at Caltech, trying to come up with some interesting problems to work on, and I stumbled upon his famous Abyssal Recipes paper, and in particular, a passage caught my eye. So he's discussing the different mechanisms at work in driving ocean circulation, and he notes that the most abundant organisms in the abyssal sea, copepods, mysids, euphausids, krill, squid, and the nectonic bristlemouth are all engaged in diurnal vertical migrations on the order of a kilometer. And then the emphasis is added by me, this migration is associated with some transport of water. And so for me, as you just heard, with an interest in fluid mechanics and biology and the interaction between them, it inspired me and has inspired other people to ask the somewhat maybe unexpected question, do swimming animals mix the ocean? Now, what do I mean by mix the ocean? I should clarify. First of all, in the context of global ocean circulation, we can ask whether the animals might contribute significantly to maintenance of global ocean circulation by stirring the denser deep water upward and vice versa, stirring the lighter surface water down into depths. Can they play a role in that process? The other question we can ask is with regard to the carbon cycle, and so we'll mention carbon here. I should have put phosphorus here instead. But in these biogeochemical processes that occur in the ocean, the transport of nutrients, carbon, oxygen, can these animals play a significant role through their swimming in the way that Munk mentioned in his paper? Now, what are some of the accepted mechanisms for the card-carrying oceanographers in the hall today? You'll know that, of course, wind forcing is known to be important. The stress of the wind at the surface can drive circulation. Estimates put that power input at around 1 trillion watts of power. Keep that number in mind. The other number to keep in mind is what we call the turbulent dissipation, the turbulent kinetic energy dissipation, which is on order 10 to the minus 5 watts per kilogram of water. Now, the other process that later was appreciated, but now again is accepted, is the effect of tides. So tidal currents, for example, flowing over bathymetry, as you can see in the image here, can also lead to kinetic energy inputs, or a flux of about a trillion watts, maybe a little bit less than that, but also turbulent kinetic energy dissipation rates of that 10 to the minus 5 watts per kilogram level. Now, we can also make estimates of what's the total energy required to maintain that global circulation. Those estimates are on, with error bars, on the order of about 2 trillion watts. And so you could add the top and the bottom there and say pretty much the case might be solved in the sense that winds and tides might be sufficient to drive ocean circulation. But nonetheless, I think it's worth asking, what about this idea of animals swimming? What role could that potentially play? Here you can see a vertical distribution sketched out of what we might expect to see in the ocean. Near the surface, of course, where there's light availability is where we anticipate most of the biomass in that upper kilometer of the ocean. However, we also know that at depth, there are also organisms at the very bottom feeding on whatever organic material might be down there. And very recently, I think we're starting to get an increasing appreciation for the fact that midwater can have many more organisms that simply don't show up in a net tow, for example, or whose acoustic impedance might not allow for us to see them by echo sounding measurements. So gelatinous zooplankton in the midwater, this is an example actually quite deep, relatively recently, of a jellyfish that was observed, portend the possibility that we could have animals actually occurring throughout the depth of the ocean. Now, importantly, the question we want to ask is how much power would be associated with this? And so Bill Dewar at Florida State, who I had a chance to finally meet in person here for the first time, based on estimates of net primary production in the ocean, which is on the order of 60 terawatts, trillion watts of chemical energy, has put the potential for mechanical energy input, or mechanical power input, at about 1 trillion watts. And so that's same order of magnitude as the wind forcing that we talked about earlier and the tidal forcing as well. Now, what's more, Huntley and Zhu, a little bit later, or rather before that study, analyzed about 100 aquatic species, ranging from krill to very large animals like sperm whales. And so over this large range of length scale and mass scale, the question they asked is, what's the estimated kinetic energy dissipation rate? Is it comparable to that 10 to the minus 5 watts per kilogram that we saw earlier? And in fact, it is. And so you can see here in the table, the first column is the mass of these animals. The second column, their abundance, numbers per cubic meter. They estimated swimming speeds, and then in that next column, the swimming efficiency. And on the basis of those numbers, you can estimate the watts per kilogram, the power expended mechanically in terms of fluid motion by these organisms. And across this entire range, again, they arrive at this 10 to the minus 5 watts per kilogram number, similar to what we saw for winds and for tides. Now, perhaps the paper that I think stimulated most of the recent interest over the past, say, decade or so in biomixing was work by Eric Kunz's group. They published in Science measurements of a vertical migration in Saanich Inlet. And so on the vertical axis here, you can see the depth as a function of time in the evening. That black patch that's rising is this migrating zooplankton layer. And so at the same time as this group was rising, they were making microstructure measurements of the turbulent dissipation that was occurring in the group. And so you can see there, those little red bars are indicators of the watts per kilogram that they measure. They observed up to a factor of 10 even larger dissipation rate, 10 to the minus 4 watts per kilogram in terms of the kinetic energy being dissipated. Again, comparable and even greater in some cases than what you would get from physical oceanographic processes. So here we had this string of results that seemed to be in support of this idea of biogenic mixing. And then there seemed to be sort of a shifting of the tide. So in Eric's own group, they later found that it was difficult to reproduce those earlier measurements. And so, for example, a paper by Shani Rousseau et al., they note that based on sampling to date, turbulent production by the marine biosphere is not efficient. So they simply weren't able to see those high dissipation rates in subsequent field expeditions after that initial one. I think perhaps the most important critique, though, came from Andre Visser in Science after that initial paper, where he noted that kinetic energy dissipation alone is not what you're actually interested in if we're thinking about mixing. The kinetic energy dissipation just tells us about how much energy is being put into the water. What we want to know is what fraction of that energy contributes to mixing before dissipating as heat ultimately. And so we can quantify this with a parameter called the mixing efficiency. Now, theoretically, we can come up with a model for what that looks like, and I'm sketching this here from Andre's paper. The vertical axis has the mixing efficiency on a log scale. The horizontal axis has a ratio of length scales, L divided by B. L would be the length scale of the animal, and B is what we call the buoyancy length scale, basically telling us the distance over which there are appreciable changes in the density in the ocean. The idea being that if mixing is going to be significant, it needs to somehow cause fluid with different properties to interact with one another. And so based on a Richardson's 4/3 scaling, you can get that constant slope there that you see for most of the plot. And then we expect at some point the mixing efficiency has to plateau at some value less than 1. He takes a value of 0.2, which is sort of the nominal value that people have used. But the point here is that swimming animals, if you look at where they sit in terms of that ratio of L on B, they sit at 10 to the minus 3 to 10 to the minus 1. And so if we track that over to the efficiencies, we expect that their efficiencies are all less than 1%. So if the mixing length scale that matters is the size of the individual animals, say due to the swirling currents that they might create with their appendages, then we expect that viscous dissipation is going to impede any meaningful effect in terms of biomixing. So at this point, this is a couple of years into my assistant professorship. I've been working on the problem for about a year, and I start to get a little bit discouraged here because we have sort of negative result after negative result in the literature. And so I figured, well, I should go back to my inspiration, Walter Munk. So I sent him an email. At this point, I'm kind of feeling like it's me and Walter against the world, so he'll be able to give me some backup and figure out an argument for why this is an important process to work on. I sent him the note, and then within the hour, he wrote back. And of course, to me, that made sense. I figured he's probably as eager as I am to come up with some arguments. We can, start a collaboration. I'm already thinking about the papers we're going to publish, Dabiri and Munk, or Munk and Dabiri. I'll let him decide. But then I start reading, and he says, "Dear John, it was partly an attempt at humor when I suggested many years ago that diurnal migration could lead to appreciable mixing. And I was amazed at recent papers who think this is not a joke." So you're laughing, but you have to imagine me at my computer, like tears welling up. But fortunately, he didn't just sign his name and that was the end of it, or else I wouldn't be here today. I'd have retired. He continued, and he noted, "People thought it was a lunatic idea when Carl Wunsch at MIT and I suggested that the moon via lunar tides could have anything to do with mixing. And now that's generally accepted. I must say we preferred the era when we were called lunatic to the era when people say we all know." And then he finished with the best advice I've ever gotten in my career. He says, "I can only say, work on problems you most enjoy. Strange things can happen underway." And so with that feedback, I decided let's press on. There's a lot of interesting questions here. And in particular, we wanted to first of all figure out a way to measure directly the mixing efficiency of animals swimming in the ocean. And then secondly, to figure out what are the physical mechanisms at work that lead to whatever measurements we see. As you heard in the introduction, our lab, for a variety of reasons, had been interested in jellyfish already as a model system for biological propulsion, for example. So here you're seeing jellyfish in Palau, in one of the lakes there, where you have these animals in some cases, hundreds of thousands or millions swimming at the same time. And we thought this would be a great opportunity to measure these processes. Now, one of the things we can do to study this is to dive with them. And Kakani Katija was a graduate student. First, she actually got her undergraduate degree in aeronautics and astronautics at University of Washington. So another kindred spirit. She's a rocket scientist, but also got her PhD in bioengineering, and then in her spare time, she was an alternate for the US Olympic figure skating team. So slightly more impressive than my hobby of fantasy football on the weekends. So Kakani today is a principal engineer at MBARI, the Monterey Bay Aquarium Research Institute. But for her thesis, the task was to figure out a way to quantify the flows around these animals in situ. We already knew we could take some of them into the laboratory and make quantitative measurements. But in many cases, these animals behave differently when they're in the laboratory than they do in their native environment. So first we can do some work just looking at the flow based on dye that we inject around the animals. And the key feature I want you to note is that each time the animal pulses, it creates what we call a vortex ring. It's almost like a donut that you might blow smoking a cigar, for example. And so as you heard earlier, those vortex rings become very important for the propulsion of these animals. But that's generally just a qualitative visualization of the flow. We wanted to develop more quantitative ways to characterize the flow. And so she developed something we called the Self-Contained Underwater Velocimetry Apparatus, or SCUVA. In retrospect, it's not the best acronym you could have come up with, but it seemed clever at the time. The idea, though, is that a scuba diver can make velocity measurements using this device in the ocean. At the upper left, you can see some early tests in the swimming pool down at Caltech. The image I really want to point you to, though, is at the lower right-hand side, where it looks like just a line of green. And that's because it's actually creating a laser sheet. So we're looking at the top of this laser sheet edgewise as someone measures some benthic flow there. I thought that rather than me describe this, I should give Kakani the opportunity to. So here's a short clip of her describing the technology at a TED Talk that she gave.
And so a single scuba diver can go anywhere from the surface to 40 meters or 120 feet deep to measure the hydrodynamic signatures that organisms create. And now I want to, before I begin, I want to kind of immerse you into what these kinds of measurements require. So in order to work, we actually dive at night. And this is because we're trying to minimize any interactions between the laser and sunlight. And we're diving in complete darkness because we do not want to scare away the organisms we're trying to study. And then once we find the organisms we are interested in, we turn on a green laser. And this green laser is actually illuminating a sheet of fluid, and in that fluid, it's reflecting off of particles that are found everywhere in the ocean. And so as an animal swims through this laser sheet, you can see these particles are moving over time. What you're going to see is that on the left, these are particle images that shows the displacement of fluid over time. And using that data, you can actually extract what the velocity of that fluid is, and that's indicated by the vector plots that you see in the middle. And then we can use that data to answer a variety of different questions, not only to understand the rotational sense of that fluid, which you see on the right, but also estimate something about energetics, or the kinds of forces that act on these organisms or on the fluid, and also evaluate swimming and feeding performance.
So altogether now we had a technology that would allow us to, with very high spatial and temporal resolution, map the flow around these animals as they were swimming. That would allow us to determine how much kinetic energy they were putting into the water. Think of that as the denominator of that mixing efficiency. Now, I won't talk about it here, but the companion technique we developed was to use planar laser-induced fluorescence. The idea is to take a dye, which has a known density that we've calibrated, and to inject that into the water. And then as the animal swims through that dye field, we can measure how the density field is perturbed by correlating the level of fluorescence with the density of that particular patch of fluid. So doing all of that, we were able to measure the swimming efficiency of the animals. And as you can see here, I've put it in a table with a bunch of other mixing processes that we typically think about in the ocean. And the jellyfish mixing was on average around 26% efficient, comparable to some of the other processes you see there, but very different from the less than 1% by order of magnitude that you would have expected based on the arguments of the scaling of the animals themselves. So there's something going on here in terms of a disconnect between what we measure in the field and what the theory would suggest. And so our next step then was to try to figure out how we could understand the physical mechanisms that were leading to this 26% efficiency when the theory said otherwise. And so when we started looking back at those videos, like the one that you see here, of course, I told you before we wanted to think about the vortex rings, and in my mind, that was the most important flow feature in the wake. Those would be the structures that would tend to swirl different density regions. But those vortex rings are only of the scale of the animal itself. So they're not going to generate any larger length scales to improve efficiency. But if you stare at these videos for a while longer and look behind the oral appendages, that rear clump that's being pulled along with the animal, you'll notice that the dye is actually just sticking there with the animal as it swims, almost being sucked behind it as it swims. You can see this here. I'll play this one more time. And then in the next slide, this is a bit easier to visualize. So the animal's going to be moving from left to right. And so you can see it draw out this wake behind it, this fluid that's being dragged along with the animal as it swims. Let that play one more time. So it's generating those vortex rings, but has this other flow phenomena that's occurring at the same time. Now, another way we can see this in the laboratory is to use the technique that Kakani described to you a moment ago, where we have a laser sheet. And so now what you can do is look behind the animal, and there's all these tiny glass beads that we've put in the water. And your eye can hopefully notice this chunk of the water that's moving along with the animal without being propelled backwards. Now, the animal, of course, can't pull itself up by its bootstraps. There is a rearward jet that's being created to generate the propulsion, and you can hopefully see that in these two alleyways on the sides. But right behind the animal, you have this flow that's being carried in the same direction as the animal. And so it turns out that this idea is a Darwinian mechanism, not Charles Robert Darwin, who most of you are more familiar with, author of things like The Origin of Species. But he had a grandson, Charles Galton Darwin, who was much worse at making titles, and so his paper is just Note on Hydrodynamics. But it turns out that this paper is really instrumental for what we're going to talk about. What this Charles Darwin thought about was the idea of a potential flow. In layman's terms, the idea here is we're not going to worry about viscosity, we're not going to worry about turbulence. This is an idealized flow model that allows us to write down equations for exactly where every fluid particle would go. And so you can see at the top there the expression for u sub r, the radial velocity, and u sub theta, which is the tangential velocity on the circle of the flow. And so he considers what happens when an array of particles, a row of particles, are displaced by a vertically migrating object, or a sphere in this case. And in particular, what he shows in these equations is that as the particles are pushed out of the way by the pressure field, remember there's no viscosity to act here, they undergo these lateral looping trajectories and ultimately experience a permanent forward displacement in their motion. Let me let that play one more time. Now, unless you think this is just a theoretical construct, I went to YouTube and found a great video of this at work. Here's the semi. And so you can see the smoke that he vapes and blows out creates these looping trajectories. Now, again, I caution you against trying this yourself, or you'll end up demonstrating natural selection for us inadvertently. Okay, so now let's just plot the vertical displacement of each of those points rather than the looping. So we're only looking at how they're displaced vertically. And what you'll find is that that row ends up tracing out this horn-like structure, which Darwin called the drift volume. So it's the volume between the initial flat surface and this conical region in which flow is permanently displaced upward. And so the interesting thing that he notes is that the ratio between that drift volume and the body volume is a constant that depends only on shape. It's the added mass coefficient. For those of you from naval hydrodynamics background, you'll know this in the context of ship design. It's the same idea. For a sphere, the added mass coefficient is 0.5, so it means that as an object moves vertically, it carries with it a volume of fluid that's half of its own body volume. Similarly, for a cylinder, you get a different coefficient. But the important thing I want to emphasize here is that at least at this point, this drift volume only depends on shape, not on size. Remember the idea of turbulent mixing, size was an important factor. The very small organisms simply wouldn't be effective at mixing because of the fact that their length scale was less than that buoyancy length scale. In this case, regardless of the size of the object, it's always going to carry with it, by this potential flow mechanism, an additional portion of fluid. Now, what is the effect of viscosity? Well, one way we can think about this is by simulating, again, on the left-hand side, a potential flow. On the right-hand side, we're going to add viscosity. And in fluid mechanics, one parameter we use is called the Reynolds number. It's a ratio of the inertial effects to the viscous effects. So a Reynolds number of 100 would correspond to an organism that's a centimeter in size, swimming vertically at a centimeter per second, just to give you a sense of the scale we're talking about here, wherein in water in particular. And the thing I hope you can note from these simulations is that the flow is even more influenced in the vertical direction than it is in the potential flow case there. You can see particles even further away from the object being dragged in the same direction. It's like taking a spoon and putting it through water as opposed to putting it through molasses and carrying much more with it as it goes due to that relative effect of the viscosity. And so you can see that here for a few still frames here, Reynolds number 1 and 10 and 100. And the coefficient C that I'm showing here is the ratio of the amount of fluid that's being carried outside the object to the volume of the object itself. And you can see as we go to smaller Reynolds numbers, the viscosity on a relative basis becomes more effective, more important, and that added mass coefficient increases. So viscosity enhances this induced drift effect as opposed to the negative effect it had on turbulence. Now, for those of you who study the physics of swimming, this should hopefully give you some pause here. I've sort of played a trick in the sense that the potential flow case had a force balance. There was no net force on that object in order for it to move at constant velocity, something we call D'Alembert's paradox. On the other hand, in order for me to move one of these cylinders at finite Reynolds number, I have to have some external force that's doing it. So there's sort of an apples and oranges comparison. And the way that we decided to resolve that was experimentally. And so I had a wonderful PhD student, Jana Naroth. She came to us from Heidelberg, where her undergraduate degree was in biology. She had an initial interest in neurobiology, but I convinced her to study jellyfish instead, which was wonderful. And now she's a principal investigator at a biotech startup out in Boston. But what she did was to take these very small ephyra of the jellyfish, so at Reynolds number 10, where viscous effects are going to be very important relative to the animal size, she starts them in these blobs of dye and then lets them swim, and so essentially does in the lab the type of thought exercise that Darwin did in his paper. And so it'll come into focus here in a moment. First of all, you can just sort of admire the beauty of these things as they're swimming through the dye. But the other thing to note is how far this animal is carrying the fluid that was initially in that blob at the lower left-hand side. So as it carries it through, both by the pressure field, but now also because of the viscous effect, it can carry fluid much further than its individual body size would dictate. Here, as I've indicated, the length scale of the transport is much larger than the length scale of the animal itself. And so if we think back to Andre Visser's plot of a mixing efficiency, instead of looking for the size of the animal, we really want to look for this length scale of the transport, and that can resolve the apparent disconnect between the low mixing efficiencies in theory and the higher values that we were measuring in practice. And so what we imagined is that in the ocean, you might have a system that starts at depth, a group of organisms that start at depth and rise vertically, and carry with them the physical properties, the salinity, the phosphorus, the other nutrients that are important for biogeochemistry with them as they rise vertically. And the other thing that we noted from some later simulations was that this effect is even further enhanced when you have groups of organisms rising together. And then we imagine that it's certainly going to be the case if I take something that's heavier and I lift it up, eventually it's going to want to overturn. And the length scale of that overturning would then be much larger than the individual animals. Again, potentially offering a way in which the efficiency of the mixing could be enhanced. And so we published this a few years back, and it led, I would say, to more questions than it did answers, in the sense that you could follow this up and ask, in which context would this potentially be relevant? We study jellyfish, but these particular jellyfish in Palau are not the most numerous in the ocean. What would happen in other locations, not in a place like Palau, in this sort of unique marine lake? What properties of the water in terms of their transport would be important, and so on. And then the sort of big picture questions that are more speculative, but I think interesting to think about. No climate model includes any sort of process like this. Would this have an important feature there? Overfishing would change the properties of the ocean in terms of which animals are there. Things like harmful algal blooms, acidification, eutrophication, all of these processes could affect the organisms, while the organisms could potentially in turn affect those processes. So complicating the feedback that occurs in the ocean. What I want to do here, though, is to think specifically about this question of organisms and to start to pivot toward animals that we think could be more important climatically in the ocean. in particular planktonic swarms of krill and copepods, for example, and to ask how these dynamics occur in those systems. So first of all, these are the most abundant animals in the ocean, and so it gives us a sense that if we see these similar type of phenomena in these organisms, then we really have a case for this being something that oceanographers writ large might want to think about. They're, as I just said, located in climatically important regions of the ocean, and they exhibit these large vertical migrations, as you saw Walter Monk mention earlier. For us, the benefit also of studying these animals is that we could use an analog of them in the laboratory where it was much more straightforward to do measurements. So we were lucky in the case of Jellyfish Lake in Palau to have the animals right there at our fingertips, so to speak. But going to the Southern Ocean and trying to fortuitously encounter one of these migrations is a much more challenging thing to try to do. So I was very fortunate to have two great graduate students who've been leading this project. The first is Monica Martinez. She got her undergraduate degree in mechanical engineering at UNAM, the National University of Mexico, in Mexico City. She got her master's and PhD in mechanical engineering and is now an assistant professor at UC Riverside. And Isabel Houghton, who got her undergraduate degree at Berkeley in atmospheric science. And again, I sort of twisted her arm and decided to work on ocean problems instead of the atmosphere. And she's been fantastic. So what Monica wanted to do was to first figure out how to study this process in the lab. And so we use Artemia salina. This is a brine shrimp, so this is not an oceanic species. But its swimming motion, in terms of the metachronal waving of its appendages, is very similar to what you would see in krill or other types of swimmers in the ocean. Their scale is on order 5 millimeters, and they're relatively easy to culture. The other thing about them that's really important, though, for the lab is they have a strong phototactic response. So what you're seeing here is a dish in which we have some of these animals. We turn on a laser, a blue laser from the top. They see that bright spot at the bottom, and you can see those specks all swim toward it and then sort of get beamed up like Independence Day or something to the top. So with this type of a process, over the course of 2 or 3 years, Monica refined a technique in which, rather than waiting for a daily occurrence of diurnal vertical migration in the ocean, we could do this on demand in the lab until the animals get tired, basically. And so this is what the lab experiment looks like schematically. It's sort of a laser light show. So I'll start on the right-hand side. We have a blue laser that's going to shine through as a single beam, and it's going to rise vertically. And that's going to be the cue for the animals to chase after and to rise vertically as well. So we're sort of tricking them into the type of motion that they undergo in the ocean, where it's thought maybe it's a cue from the moon or something else that leads to that vertical motion. There's a green laser at the top, and that's really just to center the animals in the column, because otherwise they love the blue light so much that they'll hug the side wall of the tank on their way up, and so you get lots of wall effects. And then finally, we use a red laser to visualize the flow itself. And that's important for us because we want to be able to map out what's the flow field's response to these animals migrating. And so we'll use techniques like the particle image velocimetry that you saw earlier, where the idea is that the organisms are going to swim through, and then we can measure the flow that they induce. So here's the cartoon version of this. In this case, this is initially in an unstratified tank that we did these experiments. And in a moment, let's see if this will play for us. Looks like we might be stuck up here. Let's see. All right, there we go. So they swim down to the blue laser. That's the light for imaging. That's the light for centering. We rise. We have them raised vertically, and they chase after. And so that's the cartoon version. Here's the lab setup I'll show you in a moment. Before showing you the video, I just want to orient you. So the left, you can see the image of this first tank with the lab lights on. You're going to see a double image on the right-hand side, and that's just because of the angle that we're looking at. But this initial set of experiments was just basically trying to refine this experiment of animal migration. And so here you can see plate at 2x is the vertical rising of the laser beam. And then in a moment, Monica realizes the laptop is in the way. She moves it. And so you can see the rest of the migration there. So those little specks that you're seeing in the water are the Artemia that are swimming and chasing after that blue laser. They go through that red laser sheet that you can see there in the middle. And so these were the first set of experiments where we were able to measure larger scale flow behaviors, but this was all in an unstratified environment, so the constant density in the tank. And that was, again, because we had to first figure out this question of how do we get these animals to cooperate in a laboratory environment. But over the past 3 years, then, Isabel's been working to develop a similar facility that's larger than this one, so it's about 2 meters tall, so taller and wider as well, to do the same type of experiment. And so the key question that we've been asking is, do swimming animals create these flow features, in particular during diurnal vertical migration, that's larger than their individual bodies? What I'm going to show you next comes from a paper that's currently in revision at Nature with Jeff Kostoff and Steve Monosmith as well. We're almost, knock on wood there. Referee 4, if you're in the building, come see me. I'll buy you a beer. Let's get this done. So what we first did was to use a technique called Schlieren optics to be able to visualize the wake structures that they create. And at this point, I actually want to take a moment and thank Rudy Strickler at the University of Wisconsin-Milwaukee, who took Isabel in for a week and taught her the Schlieren technique. And so just a sort of side note for collaboration, it's really been really enabling for the work that we do. So what you're seeing here is an animal that's actually swimming downward in this case, a single animal. You can see that these animals tend to create a rearward jet as they're swimming. So this is different somewhat from the dynamics that we saw in the case of the jellyfish, where you had this prominent vertical induced drift. There's been some work by Julie Yeomans at Oxford who's shown that theoretically you can get an induced drift that's either positive or negative depending on swimming mode. And so this is a good demonstration of her theoretical work. But again, at the individual organism scale, the primary features are these vortices that get shed into the wake. And then you have the effect of stratification here. So any place that you can see light or dark fringes, those are places where the initial linear stratification of the tank has been perturbed by the organism. And you can see the effect of buoyancy tending to re-stratify. Now that's at the single animal scale. We can zoom out a little bit. So what you're going to see in a minute is on the right-hand side of the screen is the primary vertical migration that's occurring. The animals themselves are going to be swimming in the middle of the field here. And so you're seeing a few animals on the side. And then we started to see events like what's about to happen, these eddies that would sort of pop into our field of view, very clearly sort of a fluid mechanical structure, very clearly larger than the individual organisms, but we really didn't have a sense of what the correlation was between these animals migrating vertically and the occurrence of those types of structures. And so we had to change our visualization technique. The Schlieren imaging was great because we could see directly the density perturbations, but we were limited in our field of view. And so then Isabel developed a really clever technique. So basically she takes that PIV idea, the particle image velocimetry idea, but just to give us a qualitative image. The idea here is that we put small glass beads into the water on the order of 10 microns. We're illuminating a cross-section, a sheet of light there, so that's what you're seeing now. And when I play the video in the moment, you're going to see animals migrating vertically during the vertical migration. And I want you to not focus on the animals swimming up. They look interesting, but don't look at them. I want you to look at these particles and see what happens to the flow as these animals swim through. So individually, they give kicks, rearward kicks to the fluid. But as this migration proceeds, you can see those kicks start to add up. And the downward flow starts to develop. And it gets stronger and stronger and stronger still. So as these animals are swimming vertically, those rearward kicks that they're giving to the fluid are adding up to the point that they're creating this very prominent downward jet. So you can imagine, again, a diurnal vertical migration. We don't usually think about the flow in this sense, but it's going to be affected by the fact these animals are passing through. This is not climbing stairs. The stairs can move. And in this case, the stairs are being pushed downward at a rate that we can visualize very clearly here. Now, we can zoom out a little bit further. Here I'm going to look to the side, because if you look in the center of the migration, basically all you see is that downward jet, which is interesting, but a downward jet isn't necessarily going to create any stirring of the fluid. It'll displace fluid that's at one height to another height. But if you look at the side here, and what Isabel's done here in this larger field of view, because you can't see the particles as well, is she's overlaying a few time steps at a time, so the individual particles almost look like little comets. but you can see the flow evolving. The animals are again swimming up, and you can start to see these eddying motions in the field, again, at much larger scale than the individual animals. The individual animal size is indicated in the red dot at the bottom there. So these animals together create this large jet. As that jet starts to re-stratify, and as there's a Kelvin-Helmholtz instability that develops at the side of the jet, you get this larger eddying motion at larger scales than the individual animals. And so one last experiment I'll show that she does. Again, in a stratified tank, all of these experiments are in a stratified tank. She has a linear stratification, and now this time what she's going to do is to put a layer of fluorescent dye at the very top, and the density of that dye is going to match the upper layer. So left to its own devices, it's just going to stay floating at the very top. Nothing's going to happen. Now we have the animals migrate vertically as we did before. They're going to generate that downward jet, and let's take a look at what they do. Again, look at the scale here now. We're at about 1/2 meter, 60 centimeters, as opposed to the 5 millimeter scale of the individual animals. So the animals are swimming up, and they create this beautiful plume. The animals are filling the full width of the light signal that we give there. So if we'd given them a wider signal, they'd have the opportunity to make an even wider system. But you can see very clearly the transport created by these individual organisms. Now, the depth to which they're propelling the fluid is constrained only by the height of the column of animals itself. So in the lab, we run into a limit where at about 50 centimeters, we can't fit any more animals in there before it's just sort of unrealistically high density. In the ocean, these aggregations can be 10, 20, 30 meters deep. And so if you're able to generate flow features at that scale, it becomes very much relevant to both biological, chemical, and physical transport in the water column. So the other question we should ask, or let me answer this question before going to the next one. Do the animals create larger features? Yes, I think we've seen that by many different modalities at this point. And you can see here a couple snapshots of the temporal evolution. And again, in the box in the upper right, I'm showing you an example of what one of these individual objects looks like. Now, does that lead to irreversible mixing? I'm glad you asked, reviewer number 4. So the question becomes whether those eddies that are created will just end up ultimately re-stratifying, or will the density profile actually be changed over time by the mixing? If the flow re-stratifies and the density doesn't change, it could still be interesting for biogeochemical transport, because those nutrients that were transported to a new location, that bacteria that was transported to a new location, et cetera, that still matters, but it may be less important for the physical oceanography piece. So in a separate set of experiments, which Isabel will describe on Friday, the answer is yes, and the mixing is huge. It's 1,000 times the molecular diffusion that would occur in the similar salt stratified environment. So cancel your flight, stay here till Friday. 10:45 in the morning, she'll go through those sets of experiments. But not only do they create the large scales, they also create very significant mixing and permanent mixing of the density in the water tank. So where do we go from here? First of all, I think the next frontier in my mind is going out in the ocean and finding direct evidence of this occurring there. Very recently, Tarling and Thorpe from the British Antarctic Survey, published a paper in December where they may have already done this, or at least gotten the first hint. So this is from their abstract. They examined vertical velocity within 2,000 krill swarms, so they're studying organisms that actually matter for the ocean, and they observed this pronounced downward anomaly of vertical velocity within the swarms. Their values are the same order of magnitude of what we see in the lab, and they hypothesize or propose that it's due to the downward velocities created by the action of swimming. So it seems like this has been seen for the first time. I think it's worth looping back for a moment to the negative results that have sort of piled up in terms of the field of measurement here. What they've measured is what I think we want to, the velocity anomaly. If I take a microstructure probe, think of the red video that I showed earlier, it's going to tell me about the gradients, the spatial gradients in the velocity that are occurring, and then I can take a shear spectrum and then integrate under that to get the dissipation. But that's not really what's occurring within the aggregation. There, it's this velocity anomaly, and then perhaps to the edges or the periphery of that group is where we might see the more energetic, turbulent effects that occur. And so the question I ask is, those here in the audience have huge data sets of, in some cases, echo data, in some cases ADV, ADCP, other CTD casts, and so on and so forth. Can we mine this to automate the discovery of at least correlations between the presence of the organisms and these physics? I think the other thing is using new in situ tools. So this is from Kakani's work at Mbari, where they're taking their rovers down to depths that we simply can't go, scuba diving, for example. And actually seeing is believing. I think hopefully after you've seen those videos in the past couple of slides, you can see how important it is to get visual measurements of the flow and not just point-wise sensor readings for what's happening. And so can we use these new tools and many others that are being developed that we're hearing about at this conference to study DVM in real time in the ocean? I think that'll be essential. And then finally, there's the question of biogeochemical signatures. So bringing back to Claudia's excellent talk, are there ways that we can study those effects and use them as a proxy to understand either present or maybe even past biomixing events that have occurred, not based on changes to the density structure of the ocean, but by changes to the chemistry, to the biology that have been induced by these types of fluid mechanical effects? So with that, I have several people to acknowledge. I've mentioned most of them over the course of the talk. Sean and Jack Costello have been some of my longest standing collaborators in the area of jellyfish research, and we've just had a ton of fun doing it over the years. Many others that are almost too numerous to mention, but I want to make sure I mention other members of the lab group here. Valerie Troutman will be giving a poster on Thursday here. She's developed a new technique to do the same type of scuba measurements, but in 3D. And so that opens up a lot of avenues for not having to, for example, balance your laser sheet right on the organism to do measurements in daytime. Christy Schluter-Cook is giving a talk on Thursday morning as well. She's developed an unsupervised machine learning algorithm that allows us to take fluid dynamics measurements and help them to be assimilated into models for more accurate data modeling. Many other collaborators, I mentioned Rudy Strickler, Monty Graham, who took a lot of the cool photos that you saw with Kakani. Eckhart Mayberg at UCSB has been an important collaborator in thinking about how do we start to numerically simulate these effects. The scales at which we do modeling, even on a regional basis in ocean modeling, is huge relative to these organisms. And so how do we achieve a bridge in terms of scaling? That's an unsolved question. I do want to thank also funding. The U.S.-Israel Binational Science Foundation took a leap of faith on us very early on in this work in funding the project when some of our NSF reviews were just like laugh emojis, basically. And so it's been really helpful to get their funding, but subsequent funding from NSF and ONR has also been essential. So let me finish by just leaving you with this great quote. If you take nothing away from this talk, except this quote, then I'll feel like it's a success. I think it is great to work on problems you most enjoy and know that strange things can happen underway. Thank you. Great. Oh, hi.
Wow. Wonderful talk, beautiful work. Thank you so much for that. I think you did justice emphasizing the damage that Visser did to these ideas with that paper. One of the other things that I've wondered about is that a lot of the animals that we were talking about were not necessarily that small. They've been working with very small animals here. But a lot of the zooplankton resident in the not so shallow parts of the ocean can get considerably larger than what you've been working with, to the point of being almost small fish that travel in large schools. And if I understand some of the physiological data that's been obtained in schools, the animals in the inside of schools tend to work less hard than those on the outside, which makes me think that there must be some sort of large school-sized organized flow the school is developing for that to take place, which again is a much larger scale, and now with much larger animals, of which the biomass is considerable. Right. So it seems that there are ways that one can expand these studies to include broader branches, other animals as well. And with that, we might find more evidence of other things to look for in other parts of the fluid. Is anybody that you know of, yourself included, thinking about this or working on these ideas?
No, it's a wonderful point. In fact, I would argue that for those larger organisms, fish, and even if you get to the scale of whales that might move together, in many respects, it's a much easier fluid mechanics argument to make that you're going to see significant effects in terms of the fluid transport in those cases. And so, in a sense, we started from the smallest organisms because we figured that was where a lot of the skepticism lay with those animals, that if you just took a straight scaling argument, you'd say there's no way they can do anything important. But for those larger organisms, you're absolutely right. That would be another group that you would expect to have significant transport. I'm not aware of people who are looking at the biome mixing implications of those groups. One of the challenges becomes finding them. So again, if you want to do this in an in situ sense, to be able to know where they are and to do the perfect experiment is kind of like what Isabel does in the lab, but imagine you could do that in the field where you're able to measure the animals. She has a density profiler so she can see what's happening there and then measure the flow at the same time. If the people developing platforms could find ways to do that and have them track these groups, that I think would be wonderful. I think that's low-hanging fruit. Yeah.
And one last comment, and that is I hope we're going to be able to sit down and have a beer and talk more about this.
Oh, okay. I am Spartacus.
I'll even buy.
Thank you. Yes.
That was a fabulous, fabulous talk.
Thank you
so much for it.
Thank you.
My question, I'm going back to the smaller organisms. My question is, are your Reynolds numbers small enough that that this mixing might mix things like temperature and salinity and whatever at different rates?
So temperature and salinity we would expect would be mixed irreversibly at different rates, but mainly because the molecular diffusivity is 2 orders of magnitude different between them.
Yeah, but that's really important for ocean, for putting that into ocean models.
Absolutely. So I think if this had been a thermal experiment, the mixing would be even higher, actually, because of those faster rates of molecular diffusivity. So at this stage, sort of going back to Bill's earlier question, we were trying to study the cases that are a bit more difficult to prove. So the salinity case, it's easier to get incomplete mixing because the flow could satiate and then not actually mix. I think that the thermal case is a really important one to do next. We've tried to do some limited experiments
the heat
years
loss
ago.
is hard.
But it's a difficult lab experiment to do. But you're absolutely right, especially if you wanted to convert this into a model, the differential mixing of heat and salt would be very important.
Thank you.
Yeah. Thanks. Yes, sir.
Like other people said, thanks a lot. That was really super interesting.
Oh, thank you.
And it's funny, just a little comment. When I first read your abstract before coming to the talk, it made me think of the first time I was in a canoe race. And you launch a single canoe and there's not much of disturbance, but you get like 20 or 30 people. And I remember just being amazed at the waves that were kicked up and the amount of energy in it. Obviously, it's not the exact same dynamics.
Oh, but similar, yeah.
But kind of a similar idea. But my question was, and it's probably a pretty basic question, is when you're doing these lab experiments with where you have boundaries on the side.
Yeah.
How does that? I'm guessing that has to interact with all these dynamics you're talking about. How does that extrapolate to the wider? If you didn't have that boundary on the side?
Right, that's a great question. And in fact, one of the reasons why we made the switch from that first setup that you saw to the bigger tank that we now have up at Stanford was to make sure that what we were seeing was not an artifact of the boundaries in the system. So we only allow the animals to swim in a column that's in the center, and so it allows the rest of the flow to evolve as it would. And then we can check with the density probe or also with the flow that if we go out to the walls, we're not seeing any lateral intrusions or anything occurring at the extremes. So I'm confident that we're far enough away that we don't have an effect here. You could ask the question if this was a kilometer scale aggregation as you might find in the ocean, and if it's in a coastal region versus the deep ocean, would there be differences? I think there certainly would, but we haven't endeavored to try to investigate that here.
Thanks again.
Yes.
Hi, John. Great talk.
Thank you.
I wanted to ask you what you think about the spatial distribution both laterally in the ocean and vertically, because I think we can make a pretty good argument that mixing doesn't matter for the overturning circulation, or at least this part of the overturning circulation you showed. because it's low in the thermocline and we can do that geochemically. But it's harder to make that argument as you get closer to the surface, right? As you think about the exchange between the mixed layer and the thermocline. So I'm wondering, what do you think about the vertical distribution of this?
Well, I would say I think the jury's still out as to exactly where vertically we need energy inputs in order to close the overturning circulation. There's certainly examples of sources that flow over bottom topography that's quite important.
That's what I was saying, the main thermocline. You know, the main thermocline, we know that almost all of the upwelling is in the Southern Ocean because of radiocarbon.
Right.
But for the rest of the world, I mean, for the nutrients coming into the subtropical gyres, the stuff that Claudia was showing, that's a different question there. And
yeah, so I think there's uncertainties on both sides. I think there's uncertainties as to where a lot of these organisms are exactly located. So it's been papers, I would say, over the past 5 years that are getting us a better picture of exactly how deep some of these groups go. In some cases, you assume they're only going to the oxygen minimum zone. In other cases, they're actually going further than what you might anticipate, just sort of logically. And so if, for example, these organisms are able to go down to 2 kilometers instead of 1 kilometer, then that makes a big difference for the type of impact they're going to have. The other part of your question, I think, at least as I would interpret it, is also laterally, in terms of longitude, for example, where are the organisms and how that is going to impact mixing, I think is going to make a big difference. And so if I was to sort of write down on a blank piece of paper what's the ideal experiment, I think we want to find places in the ocean where there's interesting biogeochemical transport that we expect to be occurring that coexists with some organisms. It might be these smaller ones, maybe it's the larger ones that are passing through that region. And also where we could, in some way, try to rule out tidal fluxes and wind as being a contributor. So one of the reasons in Palau that we thought initially it might be an interesting place to work is that you're somewhat protected, but it turns out that you do have tidal fluxes that sort of screw with your natural experiment. But that's a long-winded answer to say the vertical distribution certainly is going to matter, both in terms of where the animals are, but also in terms of the natural gradients of nitrogen, phosphorus, et cetera. Yeah.
Thanks. Thank you. That was very interesting. I have a question. I think it was about the jellyfish example in the beginning. And I think you said your mixing efficiency was 0.26.
Correct.
And then you used the Darwin pulling along of fluid. It's really interesting. But I also wanted to ask you about kind of the von Karman Street kind of alternating vortices, and I wanted to ask if you had considered those in your calculations.
Right, so the von Karman Street that you would see, I guess the closest that you get to that is really the chain of vortices that are going to be created as the animal is swimming. Generally, we think of the mixing that they're going to create as really being confined to the center core eddies that it experiences, and those are going to be length constrained to maybe a quarter of the diameter of the organisms. So if the animal's 10 centimeters, those are going to be maybe 2 centimeters. So if you go back to that plot of mixing efficiency versus L on B, they're going to be at a very low mixing efficiency. So the only way that at least we could see in which you have a length scale that's longer is by this induced drift effect, where you're actually carrying fluid, and of course you want to be carrying it against the density gradient so that you have then the possibility of subsequent overturn in the flow. But so in the calculations we do subsequently, we're mainly looking at that induced drift effect and not something like a vortex formation from von Karman. Thank you.
So breaking waves in the bubbles they also create turbulence in mixing. Would that mask the biogenic mixing?
It's possible. So especially in the surface mix layer, I mean, I've had some physical oceanographers tell me, you know, this just doesn't matter because there's so many other things that can adequately mix in the surface layer that we wouldn't care about this. I would counter that that's not necessarily true if the contribution of these is significant, because if they migrate, they go away because of population changes, then that might show up in terms of the overall exchanges that occur there. But you're right, there are other processes like the breaking wave effects that will contribute. The other thing that we think about in terms of this Darwinian effect, to go back to the previous question, is marine snow, for example, as it sinks, is going to have the same type of induced drift effect. And so yes, you're carrying organic matter or nitrogen, phosphorus, and so on within the pellet, but you're also pulling down fluid that has different properties than from whence it came. And so that's going to lead to another dynamic that right now doesn't really get a lot of traction, but I think could be interesting.
Thank you, John.
All right, thank you. Thanks for having me. It's fun.
I would like to, on behalf of the committee, I'd like to thank our speakers. Thank you all for coming and participating here. Thanks for the questions. We hope you have the remainder of the conference is great for you, and I think hopefully there are a lot of inspired people from these two talks. So thank you, and have a great conference.
Thank you.